Hello, ladies and gentlemen, of my life science classes. This is Mr. Hescox coming back to you. We are officially into chapter 5. This is DNA and Advanced Genetics chapter. Speaking of DNA, what does DNA stand for? Well, that was in your assignment the other day. That stands for deoxyribonucleic acid. You should have your vocab words defined by now. So we're going to talk about DNA and why it's so important. We'll get moving into the reading here shortly. So the opening question here, DNA is the information molecule. What does it mean to be the information molecule? Basically, DNA is the code, the information like a computer uses to run. It's what all life runs on. This code in these bases tells every living thing what to do and how to do it. DNA is a molecule, it stores information. It can't actually do anything with the information. The information has to be read from the DNA and run by proteins and other things. But the DNA is where all of this super valuable information is. That DNA has the information to start genes, to stop genes, to do all kinds of things. Everything is controlled in the DNA. How are proteins and DNA related? Proteins are very long chains of amino acids. There are 20 amino acids, and we can talk about those later in the chapter. Those 20 amino acids can be arranged from anywhere from a few hundred amino acids in a chain to a few thousand amino acids strung together to form a chain. Well, the DNA tells the cell what order of amino acids to put together to build the protein to do certain jobs. Every single protein, and there's like a million proteins, has a special gene that tells it what to do. So the DNA itself is very much like a recipe. It's the recipe for cookies. It's the recipe for a cake. The recipe itself can't make the cake, but the recipe can tell other parts how to make the cake. DNA has a very specific genetic code. It is made up of deoxyribose sugars held together by phosphate groups. And inside, there are nitrogen bases by the names of adenine, cytosine, thymine, and guanine, A to T and C to G. Our sugars are 5 carbon sugars. I'm going to put an S in here for sugar. That are the deoxyribose sugars. They have these phosphate groups that hold everything together. I'm only drawing one side of the DNA strand right now. So my sugars are all connected by these phosphate groups. P is for phosphate. These sugars are my deoxyribose. You'll notice that on one side, they're heading in one direction, and in the other, they're heading in the other direction. This is because DNA is what we call anti-parallel. And in the center, there are bases. C that pairs up with G, and A that pairs with T. And these are held by these special little hydrogen bonds you can't really see. They kind of act like a magnet and they hold this DNA together. Now there's actually 6 billion pieces like this. Here's one I drew on the board. This is your deoxyribose. The P is the phosphate group. The nitrogen bases make up the rungs of the ladder. 3 base pairs make up a codon, and a codon tells for a specific amino acid. Each gene is a series of codons that build a protein. Before you can build your protein, you've got to know how to do complementary strands and how to replicate your DNA. To write a complementary strand of DNA, you have to go A to T, C to G. All tigers can growl. So here are some practice ones. So C becomes G, C, G, C, G, G becomes C, C, G, T, A, A, T, C, G, T, A, T, A, G becomes a C, and A becomes a T. Try this next one. What does C pair with? G, A, T. T becomes A. T becomes A. T pairs with A. A to T, G to C, A to T, T to A. Oh, let's go on. T to A to T, C to G, and A to T. I don't know if you guys noticed it, but that day in class, we watched a clip from the movie Gattaca. And the word Gattaca is actually derived from the nitrogen bases in DNA. This is a picture of Watson and Crick's model of DNA. DNA replication. DNA replication involves the unwinding and unzipping. There are proteins that cause it to unwind, just like if you could unscrew a bag tie when you twist it up. They unwind and then they unzip. And then these new bases bring in matching complementary bases so that you end up with one half of old DNA So the old DNA is on the outside. And then the new DNA is actually forming here on the inside. We must copy the DNA before a cell divides so that the new cell knows what to do. It has to have those copies of the directions for life. This is called DNA replication. The steps of DNA replication are that the DNA unzips and unwinds, the proteins match up bases to the existing bases, and eventually we end up with two new. Now this goes on in lots and lots of places, so when replication occurs, it's literally occurring all throughout the genome of the cell because it takes so long to go through 6 billion bases. But replication is super important so that the new cell gets an exact copy of the original cell using the old DNA as the template strand. This is a picture here of it unwinding. There's the new pieces being brought in, and you'll end up with two pieces of DNA. These games are built in. You guys can go ahead and play them if you wish. But that takes me to about halfway through the first section. Make sure that you understand the structure of DNA, the sugars, the phosphates, and the nitrogen bases, how those bases align, how to make complementary strands of DNA, and DNA replication. And until next time, I'm Mr. Hescox. I'll be back soon to finish this section up. Until then, be good, be safe. See ya.